In this video, we'll talk about the poly-A retrotransposons. So let's first talk about retrotransposons. Retrotransposons are specific type of transposons which can hop from one location A to location B with the help of RNA intermediate. And in this process, it requires the help of reverse transcription reaction, and ultimately it moves into a new location. Retrotransposons amplify themselves fairly quickly, and that is why they are very abundant in eukaryotic genome. In fact, 49% of the genome of maize consists of retrotransposons, whereas 42% of the genome has retrotransposons in humans. Retrotransposons are of two major categories. One is LTR retrotransposons, which are very much like a retrovirus. So they are virus-like retrotransposons. And another category that we are going to talk about today is the non-LTR retrotransposons. And they are also known as poly-A retrotransposons because they have a poly-A tail, just like any eukaryotic genes. So lines and signs, these are the two predominant form of poly-A retrotransposons. Line stands for long interspersed nuclear element, whereas sign stands for short interspersed nuclear element. Let me break it down for you. Both lines and signs are short, long interspersed retrotransposable elements. They can invade to new genomic site using RNA intermediate. Now let me tell you, lines and signs are a bit different. They are found in all the eukaryotes. Almost like 34% of the eukaryotic genome is lines and signs. Lines are actually autonomous retrotransposons, whereas signs are actually not autonomous. For hopping from one location to another location, sign actually need the help of lines. And they have difference in size. Lines are bigger. Mouse lines are actually 6,000 base pair, whereas human signs are only 248 base pair, smaller. Now let us look at the overall structure of the retrotransposon, and they look like gene because they have a poly-A tail. The 5' and 3' UTRs are common between any eukaryotic gene and these poly-A retrotransposons. Adjacent to the 3' UTR, most striking thing is they have a AT-rich region which very much resembles the poly-A tail of the eukaryotic mRNA. Anyway, the ORF gives rise to two important proteins. One is RNA binding protein, another encodes for endonuclease and reverse transcriptase kind of enzyme. So lines encodes for these reverse transcriptase enzyme, which is used by the signs to hop from one location to another location. That's why signs are actually the dependent of lines. Okay, so let's talk about how lines and signs, or in general, how poly-A retrotransposon can be integrated into the genome. What is the mechanism? So here is a line. So there are RNA polymerase that would transcribe the line because it has a promoter. RNA polymerase can bind. So this is the 5' UTR, 3' UTR poly-A tail. So this is a generic line mRNA. And this line mRNA would be actually be reverse transcribed with the help of the ORF2 products. Eventually, the protein bound to line goes to particular T-rich region in the DNA. So this target site is cleaved and a DNA RNA hybrid is formed as shown in this image. Then using this poly-T, there is a first strand synthesis. Eventually, the second strand of the cDNA is synthesized and the DNA joining and repairing happens, and thereby a line gets integrated into the eukaryotic genome. But there is a fun fact. How does these kind of like eukaryotic genome avoid the possibility of incorporating any mRNA into a random location? Because any mRNA of any kind of structural gene looks pretty similar to line mRNA. And the answer lies in these endonuclease and reverse transcriptase kind of protein. So they have selective affinity towards the line mRNA. So they preferably binds to the line mRNA and target them towards specific genomic location. But by mistake, many of the cases, normal gene sequences can also be integrated into new locations, and that creates pseudogenes. Anyway, this video talks about the poly A retrotransposons and their nature, how they get integrated into the genome. But wait a second, what is the function of these retrotransposon? Retrotransposon can have many functions. It can have mutagenic effect. Imagine a transposon hopping into a particular exon of a gene and disrupting that open reading frame. So it could be mutagenic. It can lead to epigenetic silencing. It can also act like a source of long non-coding RNA. Also, it can work like an enhancer for a given promoter. So it have multiple regulatory effects. So I hope that was useful. You can visit my Facebook page or Instagram page for more such videos. Support our channel using super thanks. See you in next video.